Okay, good. So good morning, everyone. So today I'm going to present results from our research project that has been going on since a couple of years now. This current cluster, it's basically a continuation of work that was started in the previous first and second cluster. So the title of the project is Optimizing Turfgrass Fertilization to Reduce Nitrate Losses Through Leaching. So the work that I'm going to present today was mostly conducted by my grad student, Laura Côté, who is currently in her last semester of her master's program. So she's writing her thesis based on the work that I'm going to present today. So just a brief plan, the presentation. I'm going to give you all a little bit of context as to the reason for that particular project. I'm going to briefly state the objectives of our project, go over the methods that we used, give you a little bit of info on the preliminary results that we have so far, and a conclusion with the work that's going to be continued in the next year. So as I mentioned earlier, this project is a continuation of previous work that was done between 2011 and 2017, where we did a field experiment during which we compared different fertilization strategies to reduce nutrient losses, especially nitrogen and phosphorus, from the grass fertilization into either runoff or leachate water. So what that experiment told us is that if we fertilized turfgrass, we could reduce losses in runoff, mostly phosphorus losses in runoff, by 50%. So that might seem counterintuitive, but actually, if you fertilize your turfgrass, you're gonna have a much denser crop density. So it's gonna reduce runoff volume. And whenever you have runoff, you always have some contamination by nutrients. So you have some soil particles, some leaf debris, and stuff like that is carried through runoff. So if you are able to reduce the runoff volume, you are also reducing the total losses. So that's why we observed that 50% decrease in runoff losses. However, we did observe a little bit more nitrogen losses in leachate. So leachate is really underground water compared to runoff, which is surface water, from our fertilized plots compared to our unfertilized plots. So it was pretty low. So we estimate that about 4% of the applied N through fertilizer was recovered in leachate. But we thought to ourselves, maybe we can optimize nitrogen fertilization to reduce these losses even lower than that without compromising turfgrass quality. So that was the context for this project. So the project objectives were first to measure short-term nitrate losses resulting from different fertilization strategies, so N source and nitrogen rate, and also number of applications in different soil types. We also wanted to measure long-term nitrate losses resulting from the best treatments identified in Objective 1 in different soil types. And the third objective is to validate greenhouse results from the first two objectives in a field experiment on a loamy soil. So actually the same plots that were used for the first part of this project that I talked about earlier. So a little overview of the methods that we use to do that. So in 2019 and 2020, we had greenhouse trials running. So in 2019, we had the short-term part, so related to Objective 1, where we had 84 treatments repeated 4 times for each of our 3 soil types. So we use a loam, a sand, and a clay. So in each of these sands, there were 84 treatments repeated 4 times, but we also repeated each experiment twice over the year. So we did 2 runs of 8 weeks, so for each of these soil types. Phase 2, which is the object of the webinar today, was the long-term phase and was conducted during 2020. So the original plan was to take the 20 best treatments from the first phase and monitor them over a longer period in the different soil types. We did change a little bit on that approach. I will talk about it later, but the idea remains the same. And we were able, because we had much less pots, we were able to test the 3 soil types at the same time. We also did 2 repetitions over time. So we had 4 repetition in space for each run and 2 runs over time. Phase 3 will be the field trial, and it's going to start next summer. So this is an overview of what the greenhouse looks like. That was for the first experiment, a short-term experiment. So each of these pots represents 1 experimental unit, so 1 treatment. So that's, and 1 treatment corresponds to 1 type of soil. So the stable here would be 1, let's say, sand. These ones here would be clay. Each table is a replicate, and each pot is a different treatment. By treatment, we mean 1 end source applied at a given rate and over a different, a specific application frequency. So it's either under 1 application over the course of 8 weeks in that case, or 2 applications. So the same amount of nitrogen, but split over 2 applications. The same experimental setup was used for phase 2, but with, like I said earlier, we needed less space to do that. So we were able to do all 3 soil types in the greenhouse at the same time. So before I go over treatments, just maybe a couple of words about the COVID-19. So when COVID-19 hit last March, we were we were running the first run of this experiment, and we were still able to continue collecting data. We had a special permission by the university. Our experiment was considered as essential because there was a grad student on it, and if we had to stop the experiment, she would have needed, much more time to complete her master's degree. So we were still able to go once a week, collect data. So water data, I'll show you in the next few slides how we did that, and also visual quality data, but we were not able to analyze them in the lab. So we had to freeze those samples and store them, and they were analyzed last summer when we could go back to what resembles a sense of normality. And that was, before we went back to more restrictions this fall. So the treatment that we use, like I said, the plan was originally to use the 20 best treatments from the first short run experiment and to try them over a longer term. But it was kind of hard to sometimes to differentiate, which treatment was better than the other. So we kept some treatments unique. So these were really treatments that were picked from the first series and that we classified as best regarding to, reduced nitrate losses, giving us high turfgrass quality. But we also tried some treatments in combination. So instead of using a single end source, we used 2 different end sources at different proportion each. And that would be more representative of what, a fertilizer that would be used by either a homeowner or a lawn care company, or even sod grower would use. So it's very rare that fertilizer contains only 1 source of N. Most of them will have, some part of quick release N and some part of slow release N, and that's what we tried to recreate here. So, and we also added like a side trial in the first phase of the experiment. We observed that stabilized urea, in that case UFlex, was not that efficient to reduce losses compared to straight up urea. So we wanted to see if we could replicate that first. And also there was a difference between UFlex and UMax, which is like the higher quality version of the same product. So UFlex is more marketed towards lawn care companies. UMax is mostly marketed towards golf courses. The essential difference between both is that, so they're both urea, to which you add some urease inhibitors and nitrification inhibitors. So you're trying to prevent the conversion of urea to ammonium by slowing down the microbial processes involved. And the difference between those two fertilizers is basically the amount of inhibitors in the product. So UMax contains much more inhibitors than UFlex. So we wanted to see if there was a significant impact of that. And those products have been very little researched over the last few years. So we were not able to find a lot of papers regarding the efficacy of those products to reduce nitrate losses. So we thought it was interesting to try them as a side project. So these are the treatments for loam. The same type of reflection was used for clay and sands. It's just the product that changes. So we had some treatments that were using a unique end source, some treatments that were using a combination of two different sources, and a project on stabilized urea. Same concept with sand, basically. So how did we collect samples? So every week we went to the greenhouse and applied between 250 to 300 milliliters of water per pot. The goal of that was to induce a leaching event. So the amount of water varied between different soil types. So it required much more water to induce leaching in clay compared to sand, for example. But the same water volume was applied to each pot from the same type of soil. So if we applied 250 milliliters on sand, all pots that contain sand received that amount of water. So this is the Hightech system we use to do that. So it's basically solo cups that are perforated at the bottom where we put our measured volume, and the water drips slowly in the pot in order to, we wanted to make sure that there was no water going over the pot when we irrigate it. So it was easier and faster that way. Under each pot, you don't see it here, but we have a tube like that. So leachate was collected in that tube, and water was analyzed for its nitrate and ammonium content. So we had one sample each week for each treatment, each pot, basically. We also took visual quality ratings because, you know, even if you have zero nitrogen losses, if your turf doesn't look good, it's not going to be helpful. So we wanted to find a balance between good visual quality and low nitrate losses. We also, at the end of the experiment, we harvested leaf biomass to weigh it basically and to analyze it for its nitrogen content. So result from that long-term experiment. So these are the results for the loam. So on the y-axis here, you have the mean nitrate and leaching losses in the milligram per liter. That's the average for the whole 20 weeks of the experiment. At the bottom, you have the different treatments. And in order for them to be more easily recognizable, we use a color code. So the like pinkish treatments are combination treatments, so where we use two nitrogen sources. In blue, it's the unfertilized control. In green, you have the stabilized and side project that we did. And in purple, it's the single source N. So it's a big graph and a lot of things to just basically say that we did not see any significant differences between all the treatments that we used for the loam. So that's a little boring result, honestly. But if you look at the average losses, they were pretty low. So, you know, even on average, losses were under 1 milligram of nitrate N per liter, which is, so just to give you some kind of perspective, the threshold in Quebec is 10 milligram per liter. So past that, it's like a warning that water is not, that nitrate level are too high for the water quality criteria. That being said, when you talk to people who monitor water closely, if they get some kind of signal above 1 milligram per liter, it's like a warning signal to say, oh, there's something happening there. So the good news is that with all our treatments, we were well below that 1 milligram, which would be here on average. Of course, there was some events where nitrate losses went above that 1 milligram, and that's the next step in our analysis to determine if there were some treatments that went above that 1 milligram per liter threshold more often than others. So maybe we could recommend those. But the good news is that on average, losses were very low and similar to the unfertilized control, basically. Sorry, that's my dog barking in the background. That's just, it's the same results basically, but just a close up at the first 5 weeks of the trial, where we observed where losses were higher actually. So we also saw that in our first short-term experiment with loam and clay, especially losses were high initially because these soils contains a lot of organic matter, and when you take them out from the field and put them in pots, you know, it will bring a lot of oxygen in the soil, and organic matter will be oxidized and nitrogen will be released from organic matter. So we did observe a peak in nitrogen release in the first few weeks, and then it stabilized. And that was true also for the unfertilized control. But so that, yeah, that's just to show you the first few weeks of the experiment. Losses were a little higher, but again, not much difference between the different treatments in loam. In clay, similar results. So again, very low losses on average, although we did observe some significant differences among treatments. Again, that's a close-up at some weeks. And if you look here, our stabilized urea, there was a tendency for those treatments to result in, you know, a little higher losses than the other treatments compared to, you know, unfertilized and even the other combination treatments. And that was significant. If I go back to the previous slide, you know, especially that Uflex applied at the high rate of 150 kilograms of nitrogen per hectare per year. It was not that high, but it was still higher than, you know, some of the other treatments. So we did confirm the tendency that we observed in the first short-term experiment, that stabilized N is not the best source to pick if you really want to reduce nitrogen losses through leaching. So sand results, again, the scale is not the same. So losses were a little higher compared to the other soil types, but again, still pretty low on average. So we were, again, under 1 milligram per liter. A little bit more differences between treatments. And again, our stabilized urea was not really, you know, the highest per se, but there was a tendency for these treatments to be higher than some other treatments that we use. So it's not as a spectacular difference as what we observed in the first phase of the experiment. But again, maybe those sources are not the best to reduce losses. This is just the same result, but showed in another format. So we took in clay, loam, and sand. These are just our two stabilized urea applied at the 3 different rates. So compared to the control, you know, some of them were similar to the control, but some of them were higher, especially in clay. In loam, the background noise was still high even for the unfertilized control, so we didn't see much difference. But in sand, again, you know, there was some difference, especially at the higher rate. I cannot say that Umax is way better than Uflex. If you look at those data, it's pretty similar, maybe except here, but I cannot say definitively that it's worth paying more for a product that's supposed to contain more inhibitors, because it did not translate really well into results regarding leachate losses. So a little bit of word about visual quality. So I'm not going to show you the full results, but basically we were able to obtain really high quality grass while using these different treatments. So on the top here, you have different combination treatments for each soil type. And this is the visual quality. So it's no, sorry, these are the losses at the top here. So these losses were similar to the unfertilized control in all cases, and sometimes even lower. And if you look at visual quality, it's significantly higher than the unfertilized control. So we consider generally that above 6 is acceptable turfgrass quality. And so of course, the higher the better. But we were able to achieve pretty good turfgrass quality while maintaining very low losses. So in that case, it's well below the 1 milligram per liter warning threshold, I would say. So sorry, these results are a little bit boring, as I said, not much difference. But if you think about it in a broader perspective, we were really able to optimize turfgrass fertilization in the 3 soil types. And by optimizing, what I mean is that we were able to achieve very low losses from all treatments, so under 1 milligram per liter, and that were not significantly different from the unfertilized control. So basically no significant impact on the environment compared to unfertilized turfgrass. Like I said, we're going to pursue to see if the frequency of high levels, or are there some treatments that resulted in higher frequency of high levels of N, and we were able to achieve those this low impact fertilization practices while maintaining a high turfgrass quality. So our conclusion is that it's possible to grow high quality turfgrass without significantly affecting water quality compared to unfertilized turfgrass. So next step, as I said earlier, we're going to start our field trial this summer. The soil type, the native soil types on that site is actually the loam that we use in the greenhouse. So we're going to select the 4 best treatment for the loam, and we're going to compare them to an unfertilized control. So we're going to have 5 treatments repeated 3 times over these plots. So this is the plot that we're using. Each plot has a drain in the middle, and this drain can be, so water can be collected from the drain in that ditch here. So we can monitor water volume and as well as water concentration from the different plots. So we use the plots that were built for cluster 1 and 2, but we completely removed the turf that was on top and resodded with brand new turf, just to make sure that we did not, carry over some legacy treatments effects from the previous experiment. So I'd just like to thank our different partners from that for that cluster. As you all know, the cluster is a partnership program. So some money comes from Canada, but we also need some private partners to do that. So here are they. And thanks again for your attention today. So I'll be glad to take any questions if there are some.